Hello students, today we will continue with the pathogen Salmonella. In the last lecture we have seen its classification, biochemical characters, antigenic structure, viability characters. Now our objectives will be pathogenicity, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis, treatment. Okay, let's start. Pathogenesis. The process through which Salmonella infects and affects human being in salmonellosis is different from enteric fever. Now we have seen pathogenesis in E. coli. It was different. So every pathogen will have its own pathogenesis. Okay. Next, salmonellosis. Infection starts with ingestion of the bacteria followed by colonization of the lower intestine. This colonization is followed by the invasion of the mucosal layer where acute inflammation takes place. Okay, you can understand the difference. Now, with regards to enteric fever, oral infection is followed by 2 weeks of an incubation period, after which the bacteria gets to the mucosal layer through the M cells. The infection then proceeds to the local macrophages that transport the bacteria to the mesenterial lymph nodes. There's a process through which enteric fever or enteric pathogens they work. Okay, ultimately the bacteria can spread to such organs as the lung and the liver, among others, resulting in such complications as edema, cystitis and typhoid hepatitis. Understood? Pathogenicity. Determinants. Virulence factor. Okay. Now what are the virulence factors? Endotoxin, invasins, factors involved in resistance to phagocytosis, factors involved in resistance to acid pH, and VI antigen is virulence antigen. These factors are included in the property which is causing disease. Now let's see one by one. Endotoxin. Like all other enteric gram-negative bacilli, the cell walls of Salmonella, they contain PS with an antigenic polysaccharide, O antigen and endotoxin. Okay. Next, invasins. Salmonella survive gastric acidity. They survive over there. Invasins, what they do? They are proteins that mediate adherence to and penetration of intestinal epithelial cells. Understood? Synthesis of these proteins by the bacterial cell is under the control of invasin INV genes. Understood the role? Now, factors which are involved in the resistance to phagocytosis. Now, several secreted factors enable the bacteria to survive inside phagocytic cells. Now, Salmonella is a facultative intracellular organism. Please remember. Understood? Catalase and superoxide dismutase protect the bacteria from intracellular killing by neutralizing oxygen radicals. Students, please pay attention to the lecture. Some points are not mentioned in the PPT. You have to listen carefully. Okay, notes will be provided, no doubt, but please understand what I am talking. Okay, please pay attention. Next, other factors neutralize defensins, small cationic proteins that facilitate killing of bacteria by phagolysosomes. Fine? Understood factors involved in resistance to phagocytosis. Now next, factors involved in resistance to acid pH. Salmonella are protected from stomach acid and acid pH of the phagosome by acid tolerance response ATR gene of chromosome. Understood? Next, VI antigen, virulence antigen. This surface antigen of Salmonella typhi has antiphagocytic properties. Its role as a virulent factor is not clear. Okay? Now, going ahead, clinical syndromes or symptoms. Enteric fever, typhoid fever or paratyphoid fever, septicemia, third is Salmonella enterocolitis. Okay? Now symptoms. Nausea, vomiting, abdominal cramps, diarrhea, fever, chills, headache, blood in the stool. Now see, when I was talking about E. coli symptoms, I told you that whenever there are symptoms given, it is not like Salmonella has different symptoms, E. coli has different. There are symptoms which are similar for many pathogens. So unless there's a proper diagnosis, proper testing, it is not recommended to start with any kind of treatment. Okay? Epidemiology. Salmonellosis is often caused by animal pathogens through contamination and drink, particularly poultry products including eggs. And transmission is acquired via fecal oral route. Okay? Let's see laboratory diagnosis. Sample collection. Sample is collected like blood, serum, urine, feces, bone marrow, pus, CSF. Ideal specimen is blood culture, serum antibodies, stool and urine. Okay. Microscopy. Gram staining. What happens? Gram negative facultative rod shaped bacterium are seen under microscopes. Okay. Culture. McConkey's agar non-lactose fermenting colonies are seen. Blood agar non-hemolytic 2 to 3 mm smooth white colonies are seen. Understood? I'll show you the images. Now, Castaneda's method. What is it? You must have learned it somewhere. Now, Castaneda's method of blood culture, it is adopted, which provides both liquid liver infusion broth and solid media 3% nutrient agar slope in one bottle. Now it is a diphasic medium. Broth has an agar slant on one side. Okay, understood? So when we'll understand some practical things or some technical things, I will definitely explain you this method. Okay. Last is slide agglutination test, serotyping. Okay, a loopful isolated organism is taken from the colony and then it is emulsified in a drop of saline on a microscopic slide without spreading the drop. The emulsion, it must be absolutely smooth and of medium opacity. Okay, understood? Now what happens? Clumping of bacteria in test slide occurs within a few minutes. If the antigen antibody reaction is specific, we can see. And control saline emulsion of bacteria does not show any change. Okay, control and this we check both. Understood? See, these are the images. McConkey's agar, blood agar. Prophylaxis. Again, prevention part. Practicing sanitation and provision of protected water supply, washing fruits and vegetables thoroughly, drinking only pasteurized milk, practicing good hygiene, and following food safety guidelines. A divalent typhoid paratyphoid A vaccine or a monovalent typhoid vaccine is preferred. Two new typhoid vaccines, one oral and other injectable, have been introduced after many successful field trials. Okay, the live oral vaccine is a stable mutant of S. typhi strain lacking the enzyme UDP galactose 4 epimerase. On ingestion, it initiates infection but self-destructs after 4 to 5 cell divisions and therefore cannot induce any illness. Understood what happens? When this oral vaccine is taken, it initiates some infection, but also it self-destructs it after some cell divisions. Okay, so there is no induction of illness. The oral vaccine is an enteric coated capsule containing viable lyophilized mutant bacilli. Lyophilized in the dry form mutant bacilli. Okay. The injectable vaccine Typhim VI contains purified VI polysaccharide antigen. Now you know what's the role of VI antigen. Okay. Treatment. Salmonellosis disease is usually treated supportively. Now for enteric fever, patients with typhoid fever, chloramphenicol antibiotic of choice for patients with fever despite of the risk of its bone marrow toxicity is used. Okay, ampicillin or trimethoprim sulfam methoxazole are the alternatives with less severe side effects. Okay. Chronic carriers. Ampicillin is the drug of choice, but ciprofloxacin has shown promising results. Third is the drug resistance. Sporadic incidence of multidrug resistance transmitted genetically by plasmids among the strains of S. typhi. Okay. This is the treatment part. Okay. Go through the whole lecture, Salmonella, lecture 1, Salmonella lecture 2. If you have any doubts, you can ask me. Okay? Please do ask. I know you guys will be having some or the other doubt. Okay? Thank you.